Okay, so today we will learn about some basic commands of Linux platform. For that, because since mine is an Apple laptop, so I have downloaded the virtual box, and in this you can uh, simply download the operating system, which is Ubuntu, that you can see here. In Ubuntu, you can run the Linux, or if you have the window computer, you can directly use certain commands of NGS in your window itself. So as you can see here, this is the screen of Ubuntu, and this is a Linux. We can run it here. Um, so let's learn some basic commands of Linux first. So if you want to go to some file, let's say if I want to go to downloads, then I can come to the downloads, and let's say if I want to go from outside of data this download, I need to do this thing. Sorry, cd. Okay, then you can see here, if I didn't provide the gap, it didn't take the command. So you need to provide the gap in between two dots and the cd, so that you will come out of the directory. If you want to go to your home directory, you can just go to this one, and then I think I have done something. Home directory. And similarly, if you want to go to your last directory, let's say I have gone to cd downloads, then you can put the tab commands, it will automatically come. You can come here from the downloads, you can go to, I don't know which one is there, let's say maybe we can just see what are the commands. So we have in the download section this one, so we can go to the logo, let's say, and then what it is showing here is dnovo is a directory. Let's see what is there in this one. Okay. So maybe we can go to this downloads. Okay. So maybe we can go to uh, cd, will come a type command from this. Then you can go to downloads, and then you can go to this one. Okay. Now if I want to come out from this RNA-seq data, then I will just go cd, I will just go to this one, then I will directly come to the downloads. I came out from this folder, and again if I want to come out from this download, I can go to here. I can come out from here, I can come out from here, and then again I can go to from this folder. Then again I can type here and I can come to my home directory. So what are these commands actually? If you want to go to certain folder, you just type cd. Let's say I type downloads, just skip the, press the tab key, you will come in this. Again if you want to go to some other, let's say you want to go to this one, you will come out of this. Then you can just, if you want to commit out from this folder, just press 2, and then if you want to come to your home directly, just come here and do this. So this is all about this, okay? And again if you want to go to downloads, you can again go to downloads, and if you want to come to your last directory, then your last directory is this one, okay? Or you can even check it, you can go to downloads, and you can go to Salman, and then you can go to your last directory. So it will tell you your last directory is downloads. So that's how you can use certain commands. For example, if you want to create some file, let's say gedit file, you can use this command and create some file, let's say new.txt. So your file is going to create and you can save it. Right now I'm not saving it. Then if you want to move certain file from some portion to some other portion, let's say MB file, then you can, for example, this is your file name. Um, let's say sequence. You can just rename this file, let's say this one. So you will have your file name, which is sequence. Now it has been changed to sequence 1. Now again if you want to change the name of this file, let's say this to sequence 2, you can just change the file name to sequence 2, and here you can see the file name is changed. So now if you want to make any directory, you can just make mk and then you can write a directory name. Let's say if I say situ, so sorry, mkdir directory, this one. So you will find the new directory with the name Vishu. So let's similarly, if you want to, let's say, create a file name Mayank, so you will find the directory of Mayank. So let's say here you can see that. Okay. If you want to know the print the directory path, you can just type pwd and you will get your directory path, which is this one. Okay, then if you want to compress certain file, then you can use the gzip command, and you can see your file is currently in the zip folder. Now okay, now if you want to unzip it, just type you can type gun zip, and you can type the name again, this one, and then you can see this file is unzipped. So these are certain commands which you can use. Just that you can see here, this is now unzipped command. Okay, now you can also compress the file by using command eject2, and then as you can see here, it is compressed right now. And then you can again use the command and then again it is unzipped. So these are certain commands which you can use. Also there is one command which is like ctrl, and if you type this, you can go to the beginning of cell. Okay. Similarly, if you type control and F, you will go to the end of line. Similarly, if you write clear, it will clear your screen. Okay. So these are certain commands. So the commands which we learn is cd, this one, which will go to the home commands. Then if you want to go again to this file, we can go here. If if you want to go to certain other file, we can go here. If you want to come out of this file, we can just do this one more time, or we can, if directly we want to go to the home directory, we can go here. If you want to create a file, we can do G edit, and whatever the file you want, you can create it and then you can save it for you. As of now I'm not doing it. Then the bgp command we learn for the compression, and you can repeat this video, you can learn it. Now if you want to download some software, you can just do this command and use like let's say Fastqc. So if you will do here, it will run your command. Okay, it will ask your password and then it will run your command. But as of now I'm not doing it because it's already installed in my um, computer. Okay, so now we will learn about the Fastqc. So here you can see we have some certain sequences which we want to analyze. Then we have the annotation file which is in the GTF format, and then we have the reference genome which is this one. So three files we have. One is the reference genome, one is the GTF file, and one is the input file, let's say control and the treated one. Now we want to see, we can open the terminal here itself, and we can directly go to this site. Now I'll just delete it because okay. And then we can write Fastqc and whatever the sequence we have. And we can just run the command. So you can see here so we have done something else. We have to do Fastqc control1 dot, and then your analysis is going to be completed for the control side. Okay, and then you can see this sequence here. You can see everything is very pretty much good here. This is the per base sequence quality. It is pretty much good. If it is not good, then we need to cut the sequences. Then you can see the per tile sequence quality. It is also good. And per sequence quality score is also good. It should be in between at the last portion only. Then per base sequence content, it is always wrong in case of the RNA basically. And why it is so? Because what happened, in the RNA, what happened, percentage A is equal to not percentage T, or percentage G is not equal to percentage C for initial 10 to 15 nucleotides. So it is very common. This is, you are going to always get the right mark out of it and you can ignore it. In case of the DNA, you will not get these kind of stuff, you will get the straight line. But in case of the RNA, because since the percentage A or percentage T is not equal, it's not uniform in the initial 10 to 15 nucleotides, they are non-uniformly distributed. So you cannot say percentage A is equal to percentage T or percentage G is equal to percentage C, and therefore you will get this kind of zigzag motion like structure. You need not to worry about it, even though if the system is showing you the right signal. Here you can see the GC content, which is quite good. This is a per base and content. Suppose if you are getting an error, something like that, let's say a peak at 24 position, it means 20% of the bases, in 20% of the bases at 24 position, a particular base is missing. So it should not be the case. Otherwise, there is some error in your sequencing run, because your machine is unable to call the base, is unable to identify the base. And let's say the peak is up to here 20. So in 20% of the reads at 24 position, the machine is not able to identify what is the base, correct base. And that is a sequencing error. But right now there is no sequencing error, so it is good. This is the duplication label. You can see you are not seeing any no over-representative sequences. You are seeing adapter content is okay, and the keyword content means some 7 to 8 bases you are getting at the initial position in the reads. So this is a keyword sequences. Keyword means a very small number of bases are there. So this is like a short nucleotide length, which is default is known to be 7, starting at each position along the read. So here you can see here the Kmer is 20 nucleotides long. No, it's the blue one. No? Blue one is G double A double T, so 7 nucleotides long, and in the 20% of the reads it is there. And these Kmers maybe you can get because of the DNA sharing as well, if these are at the initial sequences. But since this is the orange color, so you need not to worry about it. But usually they are the 7 nucleotides bases long sequences, and sometimes they are at the initial start of the read, sometimes they are in the middle of the read, and these can be due to the sharing of the DNA. So like this, you can see the count is in 25 number of the sequences. This particular sequence is present. Like this, you can see here how many counts. 25 counts are there. And the position is read is going to be like 6. Fifth position where you are getting a green colored one keyword sequence, which is again the 7 nucleotides long. So this is the thing. So this is all regarding the Fastqc sequence. But as you can see here, sometimes your sequences are not good. Like you can see the over representative sequences. These are the sequences which you can see here. So now you need to correct them. Okay, and how you can correct them? There is a command which we need to use for this. And we are going to see the command right now. You can just copy the sequence for now as of now. And then we can open this terminal here, and then you can type Cutadapt and -a. Then we can just paste this sequence here. Then we will write the minimum length, which is 20. You can type whatever you want to type. Then your output sequence, which you can type output sequence, and you can give whatever the file name you want to give. Let's say 4, let's say 8. And what is your bad file name? You can write q_bad Fastqc, and then you can run it. Something is wrong, I think. Minimum. So you can see here I write minimum, so it will not do this. Okay, it is showing error. Okay, let's see what is the wrong file I have done. Okay, so the file name is, again you can see it is bad2. Why it is like this? Not bad2, basically it's just bad. Let's see if we took the sequence from this file only. So now just doing trimming. So it has done right now. We need to do again the Fastqc of Qbat 6 if I'm not wrong. Let's do it and see if the same sequence has been removed from there or not. Okay, so now we can see the what is the file name is Qbat 6. Let's see what is this one. Qbat 6 this file. You can see here. You can see the representative sequence, so that sequence has been removed. Similarly, for example, if you have some bad sequences, let's say we have some bad sequences. Uh, so in this, so everything is good. But let's say if we want to further improve this basic stats, so what we can do here, we can open the terminal here itself and we can write Cutadapt minus Q, then we can write maybe 25, then we can write the name of the file, which is whatever the file name we want to give. Let's say good dot first q, and this file name is, let's say, bad 2 dot first q. So it will quality score will come till 25. So now you can do the again Fastqc of that. Okay. And then you can run this one. You can see here. So everything is below 25. So whatever the number you want to give, let's say if this particular bar has come to 14 and you want it to increase the quality score to 30, you can give to 30. So everything will come to the 30 only. All the bad sequences are going to be removed. So that's how the Cutadapt is going to function. And in the next lecture we will learn about the some other commands the cuff link and the cuff merge, and the R software, and how to differentiate the gene expression level